Opabinia regalis is an extinct marine animal from the Cambrian period that does not fit neatly into any modern animal group. It lived approximately 505 million years ago during the interval known as the Cambrian explosion, when animal diversity and morphological experimentation increased dramatically. Opabinia is of exceptional importance for understanding early animal evolution because its body plan differs markedly from those of modern arthropods, annelids, or other major invertebrate lineages, and it is therefore interpreted as a representative of a stem group rather than a direct ancestor of any living animals. Opabinia inhabited exclusively marine environments, and all confirmed fossil material comes from the famous Burgess Shale deposits. Although this locality lies in present-day Canada, during the Cambrian it was situated near the equator and formed part of a warm, relatively deep marine basin. The Burgess Shale is renowned for its exceptional preservation of soft-bodied organisms, a factor that has allowed scientists to reconstruct the detailed anatomy of unusual animals such as Opabinia, which would otherwise be unknown from skeletal remains alone. The external morphology of Opabinia is highly unusual. Its body was soft and clearly segmented, with each segment bearing broad, laterally projecting lobes. These lobes are interpreted as structures involved in locomotion and respiration. The posterior end of the body terminated in a fan-shaped tail, which likely contributed to stability and maneuverability while swimming. One of the most striking characteristics of Opabinia is the presence of 5 eyes located on the head, an arrangement that would have provided a wide field of vision. These eyes were probably relatively simple in structure, but well suited for detecting light and movement in the Cambrian marine environment. Extending forward from the head was a long, flexible proboscis, another defining feature of Opabinia. At the distal end of this proboscis was a small claw-like structure used to grasp food. The proboscis functioned as a feeding appendage, allowing the animal to probe the sediment on the seafloor. The mouth was positioned on the underside of the head and faced backward, a configuration that is rare among modern animals, and further emphasizes the distinctive nature of Opabinia's anatomy. The total body length of Opabinia was typically between 4 and 7 centimeters, making it a relatively small organism, though its proboscis added to its overall visual length. In terms of ecology, Opabinia is interpreted as a benthic predator or scavenger. It likely moved slowly along the seafloor, using its proboscis to extract small, soft-bodied organisms such as primitive worms or early arthropods from the sediment. The lateral lobes suggest that it was capable of limited swimming, but its primary mode of life was probably close to the substrate. This lifestyle placed Opabinia within the complex Cambrian benthic ecosystems, where numerous experimental body plans coexisted. Opabinia was first introduced to science in the early 20th century. Its fossils were discovered and formally described in 1912 by the American paleontologist Charles Doolittle Walcott, who named the species Opabinia regalis. Walcott initially interpreted the animal as being related to arthropods, but the limitations of early fossil preparation techniques prevented a full understanding of its anatomy. In the 1970s, a comprehensive re-examination of Burgess Shale fossils by the British paleontologist Harry B. Whittington revealed the true complexity of Opabinia's structure, including the 5 eyes and the unique proboscis, fundamentally changing scientific views of the organism. From an evolutionary perspective, Opabinia is best regarded as an evolutionary experiment characteristic of the Cambrian period. It is not considered a direct ancestor of modern arthropods or any other extant animal group. Instead, it represents a separate evolutionary branch that explored a distinctive body plan, but ultimately left no surviving descendants. During the Cambrian, many such experimental forms existed, but most were eliminated over time as ecosystems became more competitive and natural selection favored more efficient and stable anatomical designs. The way in which the Burgess Shale animals were buried, by a mudslide or a sediment-laden current that acted as a sandstorm, suggests they lived on the surface of the seafloor. Opabinia probably used its proboscis to search the sediment for food particles and pass them to its mouth. Since there is no sign of anything that might function as jaws, its food was presumably small and soft. The paired gut diverticulum may increase the efficiency of food digestion and intake of nutrition. Whittington 1975, believing that Opabinia had no legs, thought that it crawled on its lobes and that it could also have swum slowly by flapping the lobes, especially if it timed the movements to create a wave with a metatronal movement of its lobes. On the other hand, he thought the body was not flexible enough to allow fish-like undulations of the whole body. The extinction of Opabinia cannot be attributed to a single identifiable cause. It likely resulted from a combination of environmental changes, ecological competition, and the emergence of more effective predators and competitors. Its highly specialized and unusual body plan may not have been advantageous under changing conditions, leading to its disappearance rather than its transformation into another animal lineage. Fossil specimens of Opabinia are preserved today in several major scientific collections. The most important and well-studied material is housed at the Smithsonian National Museum of Natural History in the United States. Additional specimens and casts are maintained in Canadian institutions associated with Burgess Shale research. These fossils continue to play a critical role in studies of early animal evolution, and remain a key example of the extraordinary morphological diversity that characterized life during the Cambrian period.